Right. My name is Buddhi. I'm just trying to explain the basic statistics or statistics that you want during your experiments and after your experiments. So I'm trying my best to explain this without mathematics or without any equation. So I'm trying my best. So this is not a statistical lecture to explain A to Z statistics, assuming that you guys know some sort of basic stat experience and you have used StatSpace. If not, it doesn't matter. You can contact us or you can just stop me and then ask any question. It doesn't matter. So I'm going to start from the beginning with zero maths and zero equations. So again, if you have any question, any doubt, please ask. Or if you don't want to disturb, if you want to chat or type into chat, the Vincent, my colleague, is there, so he will help you. So if you can't ask, just type into chat. So we will try to help you. Is that okay? Right. So let's start. First, the most important question is why we study statistics. What's the reason? Why do we need statistics? So that's the important question that you guys should ask yourself. Why do you need to learn statistics? What's the purpose of that? What's the benefit of that? So those are the main, that's the main question that you should ask yourself. Why study statistics? So why you need to learn statistics? Not mainly, the main reason is you run experiment or you collect the data. So data are everywhere. So you don't know the meaning of data. There's no meaning with the numbers. So you need to convert that data into a meaningful manner. So from the general data, you can't make any decision. You can't do anything. So you need to convert this data into a decision platform. So that bridge is built by the statistics. So statistical analysis link your data with your decisions. So that's the main purpose why you need to learn statistics. Apart from that, you need to learn how to collect the data, basically the experimental procedure, experimental design. Then you can collect the statistically valid data. And then once you collect the data, you can transform it to a decision. So that bridge is built by the statistics or statistical analysis. So statistical techniques used for many decision making, no matter what your career. It doesn't matter you are agronomist, you are a nurse, or wherever you work. If your boss gave you a data set and asked something, or if you're working in a production line, or there's something, there's an error somewhere, and your boss asks, okay, you have to find it. So you need to understand what you should do. And then once you collect the data, you need to make a decision. So how do you make a decision? So when it comes to make a decision, yes, you need the statistics. That's why the basic statistics is really important in order to do at least the basic decisions. So to understand statistical method, if you know the basic statistical method, it's easy for you guys to make a good decision based on your collected data. All right, so that's why we learn statistics. That's the main purpose of statistics. So another question is, what is statistics? What is that? So you know the bridge, and what is that? So statistics is basically art or science of collecting, organizing, presenting, analyzing, and interpreting the data to take decisions. So it starts from organizing your experiment, and then you collect the data, and you organize the data, and you present your data, and analyze them, and interpret them. And based on the interpretation, you can make the decision. So that's the main goal of your statistical experiment. So once you run the experiment, you collect the data, and then you need to present it in the paper or your thesis. You can't just attach your data set. You need to convert it into a presenting or interpreting way, so that you need the statistics. So that's why that's what statistics do. That's what you want to do. So basically, statistics manipulate, summarize, investigate the data to help you to make decisions, right? So if you don't know statistics, but if you have data, you can't do anything. So it's just a data, just the numbers. They don't have any meaning. Right, so far you learn what is statistics, why statistics you want. And then another main question is type of statistics. There are two types of statistics. You already learned those, but I would like to explain briefly. There are two types of statistics. One is descriptive statistics. What is descriptive statistics? Descriptive statistics is method of organizing, summarizing, and presenting data in informative way. So you basically organize, summarize, and present the data. And then the second type called inferential statistics. That's the important thing that you really need. So that you need inferential statistics means you use the data to make the decisions. Basically called hypothesis testing. So you collect the data from the population, and you analyze the sample, and then you make the hypothesis test, and they run the hypothesis test, and then you analyze the data, and then you make the decision. That's called inferential statistics. So the main difference between descriptive and inferential statistics is descriptive statistics involve collecting, presenting, or characterizing. So like visualizing your data to understand what's happening, mainly to describe the data. You will see those in news articles or newspapers or other social media. But inferential statistics is about estimation and hypothesis. There are advanced statistical data analysis done that you need to use for decision making. That's what you are going to do in your thesis, your experiment. You collect the data, you do descriptive statistics, but you need the estimation or hypothesis testing to make the decision. What is happening? What are the decisions that you can make from your data set? So you use both descriptive and inferential statistics mainly in your experiments, but inferential statistics is very important because that's the one that you use to make decisions. Right. Now, the data. So data everywhere. So everyone collects the data. Everyone measure anything, you get the data. Everyone collects the data. So you will start collecting the data in a meaningful way. Then they collect, store, present the data. And then the important thing is when you're going to collect your data, all right, oh good, sorry. So data stored and presented data set. So what you have to do is, if you want in your experiment, you need to collect the data. So you need to first design your experiment in order to collect data. Once you collect the data, you have to collect and then store the data in the way that you need to analyze. So you can't just put it data into an Excel sheet and then say, oh, okay, this is my data, but you need to organize, which we call data manipulation. So once you manipulate your data, basically for descriptive statistics, you have a basic idea. Based on that, you can start the analysis. Right, statistics data. Statistical data are a collection of data. So to study a problem. So basically, you will have a research question. You suppose I will give you a research question, and based on the research question, you need to design. I think some of you have already started the experiments or about to start experiments. One couple of guys actually contacted me regarding the experiment design as well. So I know they're already starting. So you need to start collecting data in a scientific way, not just because your data is there or your colleague or person sit next to you asks you to collect. No, it doesn't work. So you need to collect the data what you want based on the problem, the research question that you are going to study. And then because why you need to collect data more carefully or design the experiment more carefully, the main reason is that is the most difficult, the expensive and time-consuming part. So collecting data, until collect the data, for example, starting from the designing the experiment, if you do field work, right, you do the field work, and then you collect the data. But if you haven't collected data, or you don't have data for your experiment, or you don't know what you collected, or you don't need what you collected, and you collect a totally different data, then you're wasting your time and money. That's why data collection is the most difficult, the expensive, time-consuming part, and then it's the highest part of your entire research projects. Once you collect the data and they're wrong, and then your entire project is wrong. So you can't make any decision, because basically from our experience, we know that some students come with your data set and see what shall we do, and I need to analyze this and that, but then we realize that, okay, we just do the postmortem of your data set and we can't do anything because you haven't collected, or your experiment design is wrong. Or basically when I ask, okay, why did you collect it? Oh, my friend sat next to me, he was collecting data, or that guy told me it's in that one. So try to understand, talk to your supervisor before you start your experiment, and discuss with your research question, and then design, okay, these are the data I need to collect. Otherwise, once you finish your time period for experiment and you haven't collected your proper data, you will be in big trouble. So statistical data you can collect, basically obtained from counting or measuring experiment or lab work, field work or lab work. There are two types of statistical data, basically primary data and secondary data. Primary data are the data you collect from the experiments. So basically, you will design the experiment and then you collect the data from the field or the lab. So those are primary data. You collect the data. The secondary data are the data someone has already collected, or it's there in the database or somewhere. So some people actually use that kind of analysis. Not everyone does primary data analysis. Some actually does secondary data analysis, which is basically you go to a database and you download the data set, and then based on the data set, you analyze your data. So that's called secondary data or secondary data analysis. But primary data or secondary data, whatever the data you have, we talk a special term called variable. So variables are the terms we always use during the data analysis. I will get back to the variables a bit later, but keep that in your mind. You need to understand the term carefully, the variable. I will get back to that in short time. Right. Types of data, other types of data. You can collect different types of data, but you need to understand the types of data you need to collect, because this is important. Types of data you have is very important, because that depends on the analysis of your experiment or statistical analysis. So if you don't know the type of your data in your data set, then it's difficult to select the proper statistical analysis or statistical method, basically the method. So please try to understand the type of the data, then you can easily say, okay, based on this type of data, I need to choose this analysis or that one. If someone asks question, why did you choose this analysis? Okay, because I have this data sets. That's how you need to understand, because the type of the analysis you choose solely based on the type of your data. So it's really important. So you collect the data from counting or measuring items. And then those data you can put it into two main categories. One is called qualitative or categorical data. So categorical or qualitative data you get from categories, basically color, ethnic groups, gender, the tutorial group, the country of birth, any number, non-numerical thing you measured or you collected, those are called categorical data. The other one is called quantitative data. So you measure something. You measure, for example, the humidity, plant height, plant length, dimensions, number of children. So quantitative and categorical data. So these are the main two types of data you want. So based on your, because some people collect the categorical data only, some people use quantitative categorical data, some collect both. So based on that, you need to choose, or you need to choose the relevant analysis. So which is really important. So if you know, okay, my data set is categorical or my data set is quantitative, all right, that's good. So then I know what type of analysis I want, what is happening with your data. So please try to understand the difference between qualitative and then quantitative data. So the qualitative data is data always numbers. I'm going to talk about qualitative data now because most of you collect quantitative data, quantitative data, the numerical value. So they always the numbers. You count, you measure something, you measure the attributes of your population. That's what you want to do. Some people actually, mainly in laboratory experiments, sometimes you count number of microorganism groups or organism units, OTUs, all this stuff. So those also numbers, or those are also quantitative data. So based on the type of the quantitative data, you can divide the quantitative data into two subgroups, entirely based on the type of the quantitative data. So one is called discrete. Discretes are basically like the counts. How many number of books, how many animals, number of microorganisms, those are called discrete. And then the other one called continuous. So the continuous, basically from the term, you know that it's like the weight or plant height. You have a continuous data. There's no one different one value. So discrete data again, and the continuous data, all quantitative data. So try to understand, okay, I'm collecting, for example, some of you basically the wildlifters, they collect data or presence absence data also. And then number of animals, number of species, number of organisms, or you measure something, the distance or weight or plant height or something like so. Those are quantitative data. All right, so how do you collect the data? There are various ways to collect data. One is the published sources. You can search, or you can find, or you can read the materials, the published sources, basically journal articles, and you can read the journal articles and you can collect the data. That's the one main source. Second one is you design the experiment. You design experiment and then you can collect the data. So that's another way. Or you can run a survey or collect the data. It's a bit different to survey because most of the time, this is different from design experiments and survey, because surveys mostly and by the social study related to social science. But design experiment, what I really mean by is you design experiment for the field or the laboratory. It's quite different to the survey. That's why I put in a separate category. So I feel it's different. And you can do the observation study. You can just observe something and then you can collect the data. So you can collect in 4 different ways. Trying to understand, read the data, collect the data from published sources. So those who are doing meta-analysis or related to meta-analysis, you definitely use the published sources. And you have a research question, so you can use the published sources. Or you have, if you need to design experiment, you need to design the experiment related to the field or the laboratory. And then then you can collect the data, or you can design a survey, or you can just observe and then collect the data. So those are the main sources for collecting data. Right, let's get back to the variables. That's the most important term, and then the most common term in statistics that you use every day when you are using, starting from the experiment design, your supervisor or the colleague will start talking about the term called variable. What is a variable and why it is important, and what are the types of variable? I'm going to discuss about that. So please try to understand, or if you guys have any question, please ask, or just stop and ask, or otherwise just put it in the chat, because from now onwards it's very important, because this is where the real terms starts. And then when someone asks, what are your variables? And if you don't know what are the variables, and then you'll be in big trouble. Right, so variables, I'm not going to give you the definition of variable, but I'm trying to explain the different variables. Then you will have experience, or you will get an idea about what are the variables. So there are basically 2 main types of variables. So in your experiment, you use 2 variable types. One is called explanatory variable. Other one is called response variable. Can anyone tell me the difference between explanatory or response variable? No one is listening to me. What is explanatory and what is response variable? Can someone tell me? No one likes to talk? Hey, Vincent, everyone can hear me, right? Oh, I'm just talking to black ties and no one is there.
No, everyone can hear you.
Oh, okay. No one likes to answer. That's why I don't like Zoom sessions.
Is the response variable just like the one that we're like trying to study, and an explanatory variable is like just any other variable, but we're not trying to study it, but it can influence your response variable?
Yeah, thanks, Henry. Thank you so much for unmuting and explaining that. Really, I appreciate it, because you try to explain response and explanatory variable. Yeah, so basically the explanatory and response variables are 2 variables that you are using. Response, so I would like to say the response variable is the variable that, in the term response, that means something, you respond to something. That means the variable that you measure, right? So the explanatory variable is the variable that you change, right? You change a variable, and then based on that, you measure the response variable. When you read stat books, some books don't use explanatory or response variable. Some books use other terms. What are they? Do you know any another term for explanatory and response? If you read journal articles, sometimes some authors use different terms, not the explanatory or not the response. You have any idea?
Are they the same as independent and dependent variables?
Exactly. Thank you so much. So they are called independent or dependent variable. Some books, some articles, some people use independent, dependent. So independent variable is the variable that you change, which is your explanatory variable. Dependent variable is the variable, also response variable. So dependent variable is a variable depends on the independent variable. So you measure the dependent variable, right? You change the independent variable or explanatory variable. So let's go to a couple of examples. You guys need to talk to me. So the one example is, does meditation help reduce stress? So what's the explanatory and what's the response variable? Yeah, Sabrina.
Sorry, there's some lawn mowing going on in the background, so it's really hard to hear. But so does meditation help reduce stress? Is meditation the explanatory and stress the response?
Yeah, because you measure the stress, right? So when you have a research question, first thing you need to understand, what do you need to measure? So things that you need to measure is your response variable or dependent variable. So in this question, does meditation help to reduce stress? So what are you going to measure? Whether the stress reduce or not. So you are measuring the stress. So the explanatory variable is meditation. You do meditation or not. So your explanatory is whether you do meditation or not. Second one, does sugar consumption increase the hyperactivity? So the hyperactivity is the thing that you are going to measure. And sugar consumption is the variable that you change, because you want to study how the sugar consumption varies with the hyperactivity. So if you are given a question, research question like this, your explanatory or independent variable is your sugar consumption, and then the response or the dependent variable is your hyperactivity. So what you have to do is you give like a series of sugar levels, and then you measure the hyperactivity relevant to the sugar. So your explanatory variable, the variable that you change. So here you change the sugar consumption, and then you measure the hyperactivity. So that's how it works. So can eating yogurt a day cause people to lose weight? In this case, what do you measure? The weight loss. And then what's you are going to manipulate? The variable that you manipulate is the yogurt, amount of yogurt. So to change the amount of yogurt, 1 yogurt, then 2 yogurt, 3 yogurts, and then measure the weight loss. And weight loss is the response variable. And then the yogurt is the explanatory or independent variable. And do metal tags on flippers reduce the life expectancy of penguins? So there's a study that you put the metal tags to study the penguin behavior, life expectancy of penguin behavior. So what you do is you use the metal tags, different metal tags, and then measure the life expectancy of penguins. So you measure the life expectancy, which is your response variable or dependent variable, and then the other one, the metal tags, is explanatory variable. So is it clear? So this is really important, because this is the beginning of experimental design. So you need to understand what's my explanatory variable, what's my response variable. If someone asks, okay, what did you measure? What's your dependent variable? Oh, you don't know, right? So please try to understand the difference between explanatory and response variable. Another important term that we study in statistics, another important two terms that you need to understand are population and sample. So what's a population? What's a sample? So the population includes all the individuals or objects you are interested in, right? And then the sample is a part of that. So in your experiment, you study the sample, you collect the samples from the population, we call sampling. And once you collect the samples from the population, and you collect the data from the population actually, which is the sample data, and then you run the statistics. And then if you run the inferential statistics, which I explained you before, which is hypothesis testing and decision making, you apply that decision that you made for the sample to the population, right? So that's the difference between population and sample. So basically, if you want to study the amount of sleep that all the UQ students, amount of sleep, the number of hours that UQ students sleep a day. So your population is UQ students, right? But you can't survey all the students. So you select samples, which is called sampling. And based on the sample, you made the decision. And then based on the sample decision, you tell, oh, okay, average UQ student sleeps this much of time. So that's how you know. So in your experiment, you study your sample, you collect the sampling data, and then you infer into the population. That's how the experimental. So the statistics, inferential statistics comes to the second part, that you infer, you made the decision from sample to the population. If you collect wrong data, if you collect the sample, if you select the wrong sample, if you collect biased samples, I will get back to that later, then your decision is completely wrong. That's why the blue arrow from sampling, which is the experimental design and collection of data, the blue arrow from population to sample, which is really important, because if that process is wrong or it's not representing the population, your the yellow arrow, your inference or your decision to the population is completely opposite, right? So that's why this is important. So this entire picture actually basically explains what you have to do during your experiment. Try to understand your population and then get the sample and then do it. Right. So you already learned what's a sample. It's a subset of the population. You collect the subset of the population, and then you apply inferential statistics or hypothesis testing. And then based on that, you made the decision. All good? All clear?
Yep, sounds good.
Cool. Right. Sometimes when you read the articles or when you read the stat books, you may need to read stat books, or you definitely need to use a statistical program. You will see another couple of terms that I thought that might help for you guys. When you use the sample or the population, you will see a term called parameter, right? So we use the term parameter, which are, those are the descriptions for population and then statistics for sample, right? So what you do is you do statistics and then you try to understand parameters. So even though you cannot understand these two terms, try to understand what is happening in blue arrow and in the yellow arrow, because that entire picture is your experiment. If but the blue line is wrong, the yellow line is completely wrong. So if you want, some of you may need to do PhDs or masters, and you want to publish your results. So in international journals, so you cannot publish your results if this process is wrong. If not only the statistics, the sampling error, the experimental design is wrong. So then when people read your experimental or the method, they can understand, okay, there's something wrong somewhere. So we cannot accept it. So you cannot publish them. So if you want to do a PhD or master, or you want to continue your career with the research, or even maybe in your industry that you want to continue with like an industry, and you are given, you will get a data set and you have to do it, or you are asked to collect the data set. Then how do you collect it? And you need to make a decision. Your boss will tell you, okay, there's an issue in production line, there's an issue in the farm. So try to understand it. There's an issue in that pesticide. What's wrong with that? So you need to understand, okay, this is what is my population? How do I do my sample? And then how do I do the decision? If you made the wrong decision, the company you are working in big trouble. So boss definitely blame you. So try to understand the population and the sampling and statistics. All right. So basically, sampling and population. So population is your entire bowl of soup, and then whatever the things that you taste somewhere, it's your sample, right? So if you collect the sample correctly, you taste correctly, you can interpret your soup, right? If you haven't tasted correctly, you can't interpret your soup. You can't say anything, right? So you will explain in different way, right? People know the soup. People know how to make it, but you are going to explain it, and you explain it in a different way or wrong way, then your research will be rejected. And then another term, census, you all know the difference between sample and then the census. Right. Now, the important thing. Now you know the population and then the sample, and then how to collect the data from the population. But if you don't collect correct sampling data, you have an issue with your data set. So what's the problem? Issue is the sampling bias. Sampling bias is very important. Sampling bias occurs when you collect that depends on how you select the sample from the population, right? Always sampling bias occurs depends on the method or the technique that you use to choose from the population, right? So what you always need to do is you need to minimize the sample bias. You can't zero the sampling bias. Theoretically, yes, but you need to reduce or minimize the sampling bias. Sampling bias can be reduced by selecting the proper samples from your population. We call representative sample. So what happens if your sample is biased? If your sample is biased, the one arrow, blue arrow, the decision that you made is you cannot generalize it. You cannot make the decision about your population. So when the sample bias is exit, you cannot trust your decision. People don't trust the decision if the sample is biased. So always try to minimize or reduce the sampling bias. So if you have a biased sample, then even your supervisor won't happy, or the thesis reviewers won't happy, and you were not happy because you get the harsh comments from the reviewers. Right. So what do you do for bias sampling? What do you do? How do you minimize the sampling bias? You need to correct the random sampling. So to avoid the sampling bias, there is a method called random sampling technique, random sampling method in statistics. So if you use the random sampling method, you can reduce the sample bias, right? So take the random. So when you read the papers, journal articles, in the method part, you always see we collected the random sample, always you will see the term random sample because it is really important to mention that or run the experiment that you collected the sample randomly, the random sample. So it's not a bias sample. There are various methods for sampling, random sampling methods, like a 5-6 methods, right? You will see like a simple random sampling. That's the one main, the common one, most common one, simple random sampling method. So you each sample, same size, you collected the same sample from the population, has an equal chance of being selected. So you don't give a favoration for each sample, you randomly select, give equal chance to select from the population. This is called simple random sampling. Another one goes stratified sampling, you divide the population. If you have basically simple random sampling, if you have an even population, you can do simple random sampling easily. But if you have, what if you have uneven distribution of your population, especially the field studies, you won't get the even distribution of the population. So then you need to understand the other method, which is one is called stratified sampling. You divide the population into different strata. And then the other one is cluster sampling. You cluster the sample and then select the cluster. So difference between stratified and cluster is, in the cluster sample, you select the cluster. While the stratified, you select the sample from the strata. And then another one is called systematic sampling. That's another method. You randomly choose one number, and then based on that number, you choose the points. So I'm not going to explain each and every step separately. If you want, you can, because you may need those for your experimental design, and me and Vincent is here to help you guys. So you can make an appointment and come and meet us. So we will explain you how you can organize your experiment, so then you can minimize this sample bias. Or you can email, we have our email addresses at the bottom of this presentation, so you can email us. So those are the main methods for random sampling to reduce the sampling bias. There's another type of variable which we usually don't talk, but when we see an experiment, we can see this variable more often, which usually students don't even think or don't even consider. This is called confounding variable. Confounding variables are additional variables that can affect your both independent and dependent variable. So those are the variables. Confounding variables are the variables that you are really not interested in, but there is an effect on your both explanatory and response variables. So trying to understand confounding variables and then minimize the confounding effect is very, very important, right? So just because you collect the explanatory and then response variable, and then you try to link between the explanatory and response, yeah, that's pretty cool, but there are other variables that can change, manipulate your explanatory as well as your response. Those are called confounding variables. Confounding variables always have association between your explanatory and response variable. So these are a couple of very common examples. So someone can say, okay, more ice cream sales have linked to more deaths by drowning. So someone ran an experiment and then they found, okay, once you made the decision, okay, so ice cream sales have linked to more deaths of drowning. So that's a decision, but there are a lot of confounding variables. And check the other ones, you can see confounding variables. These are not even relevant to your studies, but so once you identify your explanatory variable and your response variable and try to list what are the other variables that may affect to them. That is the important thing. So for example, you run an experiment in a glass house, right? In a glass house, right? And then once you identify, okay, this is my explanatory, this is my response. Sometimes you notice that you cannot control some variables, some external factors. Those are confounding variables. So you need to identify them. So based on that, you need to minimize them. How do you do that? How do you avoid confounding variables? So you basically need to avoid them, because if you have a lot of confounding variables in your study, you cannot make the decision. The decision is not right. So to reduce the confounding variable, the main method that you have to do is you need to randomize your experiment. All right, so you learn two main terms. One is called sampling bias and then confounding effect. So what do you need to do for sampling bias? You need to select the proper sample using a standard random sampling method. And then there's another variable that affects your decision or the experiment, which is called confounding variable. The confounding variable, to reduce the confounding variable, what you have to do is you need to randomize your experiment, right? So these two things are really important when you are going to run your experiment, right? Random sampling and then randomize the experiment. These two are really important. There is a small video, but I'm not going to run that video for you. I will upload this one to Blackboard so then you can watch it and see how the bias, the survey results bias based on survey sample bias and then the confounding effect affect your results. So you can watch that later. All right. All good. Any questions so far? So now you guys know how to collect the data, how to collect the correct data, right? All good? Any question? Or boring? All good?
All good.
Thanks. So if you have any question, please ask. So far I explained the way in which you can collect the correct data, the reliable data. So based on the standard statistical procedures, right? So once you collect the data, what do? So you need to present them and you need to analyze them, right? So to present your data, you can use two methods. You already know them. You can use the graphical representation or the numerical, the table format. So that's how you need to present them, right? So basically, graphical representation, you get the overall pattern. and then the shape, the center, how it changed. So you can do it before you do any analysis. So once you collect the data and try to plot them, right, and see whether you see a pattern, or whether you can describe something, or whether there's a shape and there's a spread, right? So try to understand. So this graphical presentation is really important to get the basic idea of your data set. So always try to get the basic idea, and then you can talk to your supervisor easily, and then based on that you can do the analysis, right? So you use different methods to visualize your data. So remember, I told you you have two types of data: categorical data and quantitative data. So, and I asked you guys to remember what type of data you have. So for example, if you have categorical data, you need to use a bar chart or bar diagram or pie chart to present your data. Or if you have numerical data, you can use box plot or the histograms, right? So once you have data, you try to understand, okay, this is my data and this is how it behaves. So then you can get a vague idea about the distribution, the pattern, the deviation of your data, right? Or even you can identify outliers, right? I will explain outliers later. So you can understand outliers. So based on that, you can do, okay, what are the other stuff I can do? Right, so categorical variables. So this is a very basic example, different phone users. So number of something, and you categorize them in the categorical bar charts. Or if you want two categories, for example, you want to compare one to another, you can use side-by-side bar graph or stacked bar graph. It's the same thing. You can stack or you can put in side by side. It doesn't make any difference, but some people prefer stack, some people prefer side by side. So if you want to compare two categories within your data set, so then you can use the clustered or the side by side or the stacked bar graph. So here, what they, in this graph, they collected data about something, don't worry, but they want to compare these with male versus female. So then you have a one segment or the cluster, which is the gender. So then you put male, female for one category. For example, in a relationship, the second category or the third category. So the segmentation or the clustering entirely depend on what you want to do or present from your data set. Not just because other people say you cannot just plot it. We have seen the graphs with no meaning. And then when we ask why did you plot that? What's the meaning? Oh no, just to see, like my friend told me, but there's no meaning. You put it in your thesis, but there's no meaning. So always the plotting is entirely depends on what you want to present, right? Here in this one, they want to compare the male-female difference. So that's why they put it. And then quantitative data visualization. So you can use dot plots, you can use histograms, right? But when it comes to quantitative data, you need to also try to understand some terms, which I explain you later. Symmetric or skewness, mean, median, and outliers, all this stuff. So I will explain this stuff one by one later. So let's go. Winston, you are checking the chat, right?
Yep.
I can see, is there anything important that we can discuss? You want me to stop and?
Just we're talking about confounding variables in experiments outside, well, environmental factors with confounding variables in experiments outside the glass house, which kind of aren't, but anyway.
I can't see I cannot open the chat, so
Yes.
you can answer, right?
Yes.
Oh, okay. Because if I open the
Yes.
outside the, okay, I can see what if the experiment is placed outside the glass house. What if the experiment is placed outside?
Well, generally, if you have an environmental factor in an experiment outside the glass house, it only matters if you're doing the trial over multiple time periods.
Yeah.
And then you kind of ignore it anyway.
Yeah.
With the blocking. So yeah.
All right. Thanks for the questions. So Vincent, just please have a look at this. Right. Graphical presentation, you can go for numerical variables. This one, I'm not going to explain this one now. I will explain the skewness later. So when you plot your data or when you see your data, what kind of pattern or behavior you have. Let's start with the box plot. That's the important thing that I need to explain. So the box plot, what is a box plot? Box plot explain, basically we call 5-number summary. So when you see a box plot, you can get 5 numbers from a box plot. We call 5-number summary. It's basically about the distribution of your data, right? So this is a typical box plot. You can see AQR, median, Q1. I will explain this now. So when you organize your, let's say you collected plant heights, right? 10 plant heights from a particular plant. And then when you organize your data, you have 10 plant size, you divide this plant heights into 4 groups, the main groups, ascending or descending order. You divide the main 4 like a 25%, 50%, 75%, right? So once you divide your data, the first 25% called Q1, the 50% of your data called Q2. We don't usually call Q2, we call median, the middle value. And then the 75% of your data called Q3, right? And then you have, in a box plot, you have minimum and maximum lines. And then after this line, or in the minimum region or maximum region, you have outliers. All right. So the first 25% of your data called first quartile, Q1, and then the 50% of your data called Q2, we call median, and then the Q3 here, and then the maximum, right? And then the difference between Q3, the 75th percentile, and then Q1 we call interquartile range. Interquartile range, right? So based on the interquartile range, you can calculate the outliers, right? So once you get the Q3 and Q1, you get the difference, interquartile range, right? And then you can calculate the distance of this distance. How do you calculate this distance? You get the interquartile range. So you have value here, the Q1 minus 1.5 into interquartile range. So Q1, this is lower than Q1. So Q1 minus 1.5 of interquartile range, you have this distance, right? And then the Q3 plus, so this is your Q3 plus 1.5 into interquartile range, you get this range. So any value beyond this range we call outliers. That's how you need to do it. In Excel, you can easily plot this one in box plot and you can see the outliers. So if you want to see outliers in your data set, you just plot it in Excel and get the box plot, and you will see the Q1, median, and Q3, and then the outliers. So identifying outliers very important, because that tells you whether you have some issues with your data, or you sometimes outliers. So outliers comes because you entered data wrongly. Sometimes when you enter your data, you mistakenly add another zero or change decimal point, right? Or that there's a wrong somewhere in that particular section. So you can easily find that, right? So when you find the outlier, you can go back to your data set and see, okay, I entered this data wrongly, or there's something wrong somewhere, or whether there's something wrong with data collection. So you can identify that. So that's pretty important. Right. So once you have box plot, another thing you can do is you can do center measurements. Center measurements are basically the summary of your data. So basically to understand, okay, what's the summary of my measurements? So there are a couple of center measurements: the mean, median, mode. We don't use mode mostly, so use mean and median. So mean, you already know that it's basically summing up all the observation and divided by number of observations, so that you get the mean value. And then median is the middle value of your data. So if you order your data set in ascending or descending order, if you get the middle value, that's your median. And then the mode is the most frequent observation, right? You will mostly use mean and median. If you check your box plot, you can get the median, or if you calculate the mean, you can get an idea about the mean value. These two are really important, mean and median, when you are trying to explain your data, because those are main center measurements that you have to use. So what's the difference between mean and median? Why do you need both? Will they change interchangeably, or whether they affect? Let's try to do a simple calculation and see whether how the median sensitive or how the mean sensitive for outlier. So this is a small example. So you have 10, 20, 30, and 800. So 800 kind of outlier. So you have this data set and you calculate the mean, and you get the mean 215. But when you have a median, so median is 25, right? So mean is what you can see, because I don't want to tell, but what do you see from here? You have a huge outlier here. So outlier influence your mean value dramatically, right? And then it change. And so if, for example, you in your data set, you mistakenly put another zero, instead of 80, you put 800. And you're trying to explain your data and your mean is 215, which is not even closer to these three, right? Not even closer. So the mean is in somewhere very closer to here, not even closer to there. So this mean doesn't represent these numbers. So there's something wrong, which is your outlier. So outlier is very, so mean is very sensitive to outliers. So that's why mean is really important. All right. Once you calculate the mean or median, you want to see, like, for example, you calculate the plant height of 5 plants, and each time you get 10 plant heights, 10 centimeter, 10 centimeters, 10 centimeter, and 10 centimeter, right? 5 plants, 10 centimeter, right? And when you get the mean, you get the mean 10 centimeter. There's no doubt. So exact value. But in reality, you won't get that, right? Some plants like 8.5, 7, 12, or 10 values vary. So when you calculate the mean lies in between, but it's not entirely correct with the previous example like 10, 10, 10. So you have a variability of your measurements, right? So you need to understand that variability of your measurements. How do you do that? You need to calculate the very basic stuff called range, variance, and standard deviation, right? So if you have standard deviation, you guys heard, I'm assuming that you learn variance and standard deviations. So why you need them? You need those values because you won't get, every time when you measure the same thing, you won't get the exact value. You get the different value because you have a variability of your measurement. So to explain the variability, you need the variability measurements. Those are the variability commonly used methods, right? The range, actually, you get the difference between largest and the smallest. So you have an idea about the range. You have a highest value minus the lowest value, and then you can see, okay, my values lies between these two. So it's kind of a, we call crude of the variable, crude measurement. You can't exactly say the variability, but you can get the values lies between this and that, the highest and the lowest. That's called range. And then the most important term, the statistical term that we use every time people use is variance and standard deviation. So variance is, so you have a data set, and then based on your mean and your individual number, in our example, the plant heights of 5 plants, you have a mean and then the 5 plants height, and then based on that, you can calculate your variability. How do you calculate the variance? So you have, let's say in this example, let's say you have plant height, 3 plants, plant heights 5, 7, and 3 centimeters each. So you can calculate the mean, which is 5. And then you need the variance. How do you calculate the variance? So you calculate the variance by subtracting each number and then square them and then get the 3 minus 1, which is your how many samples? We call n minus 1. 3 minus 1, 3 number of plants, 3, then you get the number 4. So that's the variance of your data set. So variance is 4. But most common variation method is called standard deviation. Why I explain variance first? Because variance and standard deviations are pretty similar. Not similar, actually. You can calculate variance. We can calculate the standard deviation from variance, so you can calculate the variance from standard deviation. So they are like kind of interchange. So standard deviation is a term that measures exactly the dispersion of your data set, which is relevant to your mean, right? So if you calculate your variance here 4, if you square root it, you get the variance, which is number 2. What does it mean? So you have a mean here, number 5, and your standard deviation is 2. So your data set varies plus to minus 2, right? So standard deviation is 2 here, and then your mean is 5, So you have standard deviation. You can have deviation from 5 plus 2 or 5 minus 2. It's exactly which is 7 and 3. So 7 comes with 5 plus 2, your standard deviation, 3, 5 minus 2. So that's how standard deviation comes. So standard deviation is important because it remains the scale. If you measure it in the centimeters, you get the standard deviation in centimeters, right? So you can actually easily interpret them. If they are in meters, standard deviation also in meters, so it has a unit and you can easily interpret them. My deviation is 2 centimeters or 5 centimeters, depends on your standard deviations you are getting. You don't need both variance and standard deviations. Mostly common one is standard deviation because you can easily explain it, right? But people use variance as well. Okay. Right. Another variability measurements. There are other variability measurements. I'm not going to explain them in detail, but just for your information, if you guys want to learn more, or if you, when you are reading a journal article or a book, sometimes you notice those terms. So that's why I put some of these terms and I will explain them in briefly. So one is called quartiles. Quartiles are, we already learned quartiles. That's kind of a variability measurement. First quartile, second quartile from the box plot. So the first quartile explains the first 25% of your data. Second quartile explains 50%. So second quartile is between 25th and the 50% percentage of your data. And then when someone say Q2 or the 50th percentile, which is your median, which you want to understand, because when you read journal articles or papers, you will see this kind of box plots, and then you should be able to understand and then try to say, okay, these guys get the median, I'm not a couple of outliers there. So how do you interpret the outliers? How you get the median, all this stuff. So this is quite important when you are reading your articles or the papers, books. And then, like I said, explained before, you have quartiles, Q1, Q2, and Q3, and then most importantly, the interquartile range. Right, so the small example about how you can calculate the percentiles. You have numbers. I'm not going to explain. You can try later. I will upload this one into Blackboard. So one important thing you need to understand here is the interquartile range. So because interquartile range explain how you can calculate these endpoints of your box plot, and then finally the outlines. And there are other two, not main, but some fields used. Those are called deciles and percentiles. Most of the pharmaceutical or biomedical studies use percentile instead of quartiles, but other sciences like agriculture or the ecology studies use quartiles. But deciles are pretty simple. The quartiles, you divide your data set into 4. Deciles, you divide data set into 10. And the percentile, you divide data set into 100. That's the difference. So 4, 10, the 100. So that's the difference. So you can easily calculate them and get the percentiles or deciles, right? So those are just interpretation. If you want, you can ask or learn, right? Because I want to go for a very important section in your data set, which is the shape of your data, right? Shape, what is the shape? Shape, why shape is important? Why do you need the shape of your data? You need to, once you collect your data, if you have a data, you need to understand, okay, what's the shape of my data? What does it look like? How does it explain? So shape is important because that explains the variability and then how you can apply the different statistical methods. to measure the shape of the data set, there are two things that you need to understand. One is called skewness and kurtosis, right? You might have heard skewness, but not the kurtosis. But I will explain both, just in case if you read papers or the books. Sometimes you will see the kurtosis is this value, the skewness is this one. So what is skewness? So skewness is the measure of symmetry. Why symmetry is important? Symmetry is important because based on the symmetry, you want to organize, or you need to analyze your data based on the symmetry. Theoretically, you will get the data set like this. We call bell-shaped distribution, normal distribution, no skewness. So if you don't have skewness, you have a bell-shaped distribution, your mean and median lies. So your mean equals median, right? If your data is normally distributed alike, have bell-shape, your mean and median. But if your data negative or left skewed or the right skewed, your mean change interchange, right? That basically happens because of the outliers. This is mainly theoretical. You mostly get like this or like this, but not highly deviated, but similar to this one or this one. So positive or the right skewed, longer, you have a longer right tail, which is the higher, the middle value is somewhere here, the mode is here, and the mean somewhere here. So negative or less skewed. So don't worry about if you get the this kind of data or this kind of data, but if you have a lot of outliers, there's a problem with the data set, but you mostly get this or this kind of data. I have seen, but this is basically the hypothetical scenario, but I have seen this kind of data. Some students showed me, but these are the three main symmetry distribution pattern of your data to measure the symmetry. This is called skewness. So kurtosis is a bit different. Although you have symmetric Bell distribution, this Bell distribution can be changed, right? What if the bell is going up here like this and finish like this? Or bell goes all the way like this and go like that. So that's called kurtosis. So bell distribution can be like this, can be like a green line, or the red line, or the blue line. So you need to measure so if you have a normal distributed data set, the kurtosis should be 0. Then it's a normal distribution. Otherwise, it goes to higher or the flat. So that's how kurtosis work. But most importantly, you need to remember the skewness of your data. All good? Any question? Or you are listening or you're not listening? Just on this Zoom and then just went away.
No, it's all good. Making sense.
Thanks. Thank
Yeah.
you. Thank you.
Making sense.
Yeah, thank you so much. Otherwise, I always feel like I'm just talking to my monitor.
It's okay. I'm listening, buddy.
Thank you, and thanks for encouraging me. Right. Now you present your data. Then you have, now we collect the data, correct data. You presented your data. Then you need to make a decision from your data. The third part, the most important part, assuming that you collect, randomize, random sample, correct the data, and you have present them correctly. Now, the most important part is make the decision from your data, which is hypothesis testing. That's the decision part, which is one of the most important part of your statistical analysis. What is hypothesis testing? Hypothesis testing is the test to make a claim or make a decision about the population. So this test is, the results of this test is valid and then apply to the population if your data collection is correct, not biased, no confounding effects. Then you collect the correct sample data, then you can run the hypothesis test for your sample and make the decision. So it's uses for claiming about the population. Right. Hypothesis testing is a statistical test for competing hypotheses. So there are two types of hypotheses that you test. One is called null hypothesis, the term called H0. Other one is called alternative hypothesis. So the null hypothesis is basically null means nothing, right? Nothing. So claim is there's no effect or no difference. If you don't remember, just understand null is nothing, so no different. So alternative is there's something. So that's the difference between null and alternative hypothesis. So null hypothesis, nothing. Alternative is there's something. So if you have a research question, for example, our previous one, yogurt, eating yogurt reduced the weight, right? So what's your null hypothesis? There's no effect of yogurt on weight loss, nothing. And then your alternative hypothesis, yogurt reduce the weight loss. So that's how, if you have a research question, try to write your null and alternative hypothesis. So that these two are the decisions that you make from your data set. That's the decision, the conclusion you made, right, about your population. So try to understand the correct hypothesis, which one is my null, which is my alternative, and then you can make the correct claim. This is important to your thesis because your entire discussion depends on this part, hypothesis testing, right? So entire discussion of your thesis or your journal article depends on the results of this. So you are criticized or you explain with other results or others' results or others, everything with this. So that's why this is important. This is one of the main part of your research. So now you know what's a null and alternative hypothesis. How do we select whether my data is in null hypothesis section, null hypothesis or alternative? How do we do that? So that's how statistical analysis come to place, right? Statistical data analysis come to this null or alternative hypothesis. So now you have heard all the t-tests, Anova, all the tests. Those are tests that you need to do this null and alternative, right? Those are called hypothesis testing. And then based on your test, and you need to choose whether I choose null or the alternative, okay, the decision. So for that, you need to establish some decisions or cutoff, or we call some cutoff line to select whether I choose null or alternative. So that selection, the criteria to select null or alternative depends on the decision depends on something, a cute little thing called alpha value, right? The alpha value, which is the cutoff number that you have to use to choose between null and alternative. that defined. So that alpha call your risk of type 1 error. I will get back to this alpha and all this stuff later, but for this slide, I just want to tell you how you choose null and alternative. You can't choose one based on something. You need to choose which one. That is your alpha level, which explain, okay, I reject the null or I accept, I reject the null hypothesis, or there's no evidence for only. So that's how you need to do hypothesis testing. So this is the logic. So you can reject or not reject the null, right? You can reject null hypothesis or you do not reject the null, but you cannot accept the null, right? You never say you can accept the null, because why you cannot accept the null? This is really important because most students, they write accept the null. Isn't it, Wilson?
Yes. Actually, the pet hate
Yeah.
of accept the null.
Yeah. So accept the null, because you cannot accept the null because you don't have enough evidence to accept the null. So try to understand what you need to write after your decision. So there are some particular terminology, not the terminology, the arrangements of the words that you need to carefully use in order to write the conclusion based on your hypothesis test. So your experiment convince against null or statistically significant or find a favor for the alternative. That's what you need to understand, right? You cannot accept null or can say, okay, there's no effect, because there's no effect because you only ran one experiment. Like someone can run a different experiment somewhere else, other side of the planet can get the results. So then they can say, okay, we reject the null. So that's why we cannot accept null. We can say we do not have enough evidence, right? So that's how it works. One important thing I have to tell you is in this hypothesis testing is p-value. Everyone uses this term, p-value, p-value. Everyone wants this p-value, right? So when you run an experiment or when you read an article, you will see p-value. What's a p-value? Why do you need it, right? You already learned this one. So you use the p-value against to selecting a cutoff line between null and alternative, favor of alternative, right? So that p-value is very important to say, okay, there is a significant difference or not, right? So I'll get back to that. So if you have a data set, so you have a symmetric data set, and then you have a cutoff 5%. How that 5% is called alpha. We also call type 1 error. I will get back to the type 1 error later. So in your experiment, when you run experiment, you always need to understand you have kind of error which is type 1 error. So we put some sort of a percentage for the type 1 error, sometimes 5% or 10%, right? So 5% or 10%. So if your test experiment run with 5% error or 5%, you have 90% of the other side. If you have 5%, you have 95% in the non-rejection region. So acceptance region is 90 or 95. It's entirely depend on whether you choose the standard or the default methods in statistical software, or standard method is 5%. Also we call 0.05. that's a cutoff line, right? 0.05 is that 5%, or if you use 90% significance or 10%, you go 0.1. So that's how you get the significance. So if your test is under 5% significance, your cutoff probability is 0.05, right? So if you have the value, the test, the statistical test gives you a p-value, right? If the p-value is less than 0.05, your decision is go to rejection null. If the p-value is higher than 0.05, it goes to the do not reject null. Is that clear? Yeah? Probably not. You can meet us anytime, email us, and if you want any clarification, please ask, right? Because this is kind of like a 1 week or 2 week statistics in 2 hours. So I'm trying my best to summarize everything. Right, so that's how the p-value comes to the picture with the significance. So p-value is small or larger or the extreme. It's depends on whether which null, whether you choose null or you go for null or you go for alternative. Depends on the p-value. P-value is a test. Statistical test gives you the p-value, and then you have the cutoff value, which is 0.05. So based on the cutoff value and then the p-value, you can choose which way you need to go. Right? The smaller p-value, the small is how much? Is 0.05. If you, it's a standard one. If it's 95%, and then you go for in favor of alternative. If it's small or reject, it depends on 0.05. So that's why it's pretty important. You run your statistical analysis in your data set, and basically the program gives you a p-value. Everyone is p-value, so based on the p-value, you can decide, okay, my test is significant or not. If your p-value is less than 0.05, we call your test is significant. Test is significant means you have enough evidence to that you go for the rejection of the null. You have something. For example, our case, yogurt, eating yogurt reduces weight, right? Because you have enough evidence. So the alpha, also called threshold, 0.05, 5%. We always use that one. It depends on some studies, you will see 10%, some studies use 5%. Right? So why this 5% comes? That type 1 error, right? So type 1 error means the error of your statistical or hypothesis test. So there are errors in your hypothesis test. There are mainly 2 types of errors, right? Why you have errors? You have errors because you, based on the collection of your data, so you run your statistical analysis, and then you need to identify, you need to understand the types of errors that hypothesis testing need to address. So hypothesis tests entirely depend on your sample data, not your population data, right? So if your sample data is not reliable, and then it misleads your conclusion or the hypothesis. So there are mainly 2 types of errors. There are 2 types of errors. One is type 1 error, other one is type 2 error. There are 2 main errors in hypothesis testing. What's a type 1 error? Let me explain these two. It's pretty bit technical, but please bear with me. You might need those for your experiment. So in reality, H0, which is your null hypothesis, true or null is false. So you have, based on your H0, I'm just trying to explain with the shortest terms. So if H0 is true or false, it's reality. Based on that reality, you make the decision true or false, right? So there are, you can make 4 possible outcomes from this, right? One is you reject the null when it is false, right? So if you look at the first 2 boxes, like H0 is true or false, test decisions true or false. So the type 1 and type 2 errors depend on the reality and then the test decision. That's the error. So you can make 4 outcomes. One outcome is reject the null hypothesis when it is false, right? If you reject the null hypothesis when it is false, it is a correct decision. The reject the null hypothesis when it is true, that's a second null. So you reject the null, but it is true. Is it correct? You reject the null. You try to reject something true, which is not a good outcome. And then third come is retain the null hypothesis when it is false. You keep something false. That's not right. And then the fourth one, retain the null hypothesis when it is true. So that's the fourth one. So based on the hypothesis testing, the reality versus test decision, you can make 4 outcomes. Reject the null when it is false, reject the null when it is true, retain or keep the null hypothesis when it is false, and keep the null hypothesis when it is true. So within those 4 outcomes, there are 2 outcomes now wrong. One is reject the null hypothesis when it is true. So you try to reject something, it is true. And keep the null hypothesis when it is false. You're trying to keep something false. So outcome 2, outcome 3 are wrong, right? Those are errors. So the outcome 2, the reject the null when it is true, is a one type of error. And then the outcome 3 is the second type of error. So we give name for it, right? So if you look at this table, you can see if you reject the null when it is true, you have an error which is type 1 error. And if you reject the false one, it's a correct decision. And if you do not reject the null when it is true, you made the correct decision. But if you do not reject the false one, you try to keep the false one, which is type 2 error. So these 2 are the main errors, decision-making errors in hypothesis testing. And then that 5% comes for that type 1 and type 2 error. Why this is really important? Because it makes huge difference. For example, like this, right? So which one is a type 1 error, which one is a type 2 error? So the doctor made the decision based on the test results. The first picture says you are pregnant, and then the second one says you are not pregnant. Which one is a type 1 error, which one is a type 2 error? Can you see the difference? Can you see the amount of the reliability of your decisions? How type 1 and type 2 error act to your decision? So I'm going to let you decide which one is a type 1 error and which one is type 2 error, because you already know that one, these two, and then how to define these two. So you think about that and then think about your experiment and see, okay, what kind of type 1, type 2 errors that you can make. And that's entirely based on your test results. And these doctors made the decision based on your analysis. So test results, lab report. So you made the decision. So based on that. So think about the main errors, big mistakes in the real world that you can make based on these two types. Right. So you learn one basic aspect of hypothesis testing. And one second. So you know type 1 and type 2 error, and then hypothesis testing. And then one other important thing you need to understand is the confidence intervals. There's another term that you need to understand, which is called confidence intervals. What are they? I just put confidence interval at the after the hypothesis testing because I thought it might be more useful after hypothesis testing. So what's a confidence interval? You have already heard confidence intervals. And even from the term, it says confidence. So make a confidence. So confidence intervals. That means there's something good, right? Confidence interval. But I can't say good, but there's a confidence between that interval. We call confidence interval. What is that? It's kind of the range of the parameters that you can give to your data set. So confidence interval. How do you calculate the confidence interval? So why do you need the confidence interval? So I'm going to explain why do you need the confidence interval, why it is important, how you can interpret it. Okay. So confidence intervals. So confidence interval depends on the sampling distribution, and then it calculates based on your test statistics. Okay. So confidence interval, let's say you measure the plant height, like 5 plants, 10, 15, 20, 12, something like that, right? And then you get the mean, and then that mean represent your plant heights, but there is a variability. There is a variability because if you measurements, the 5 plant heights are 10, 10, 10, 10, 10, your mean is 10. Then there's no variability, right? You can exactly say 10. Then do you want the confidence interval? Because no, you don't have confidence interval because there's no interval because you have the mean 10. But in reality, you won't get that. You get 15, 10, 5, there's a variability. So based on that, that's why you need the confidence interval because you need the interval between your mean to explain confidently, this is how my mean varies. That's how confidence interval comes to the picture. How do you calculate the confidence interval? You need two things. One is your statistics, test statistics, I will tell you that later. And you need the standard error. Standard error is important. So to calculate the standard error, this is the only equation in this presentation that I put. Sorry about that. This is the only equation. I just want to tell you, otherwise it's hard. So you need to calculate the standard confidence interval. So you have the mean, you have the sample standard deviation, and then the sample size. So this section, standard deviation divided by square root of n, this section called standard error. Standard deviation divided by square root of n. And there is a symbol here, C, right? This depends on the distribution you are interested in. Sometimes you run Z distribution or T distribution, right? So standard deviation divided by square root of n, this whole section called standard error. So if you go back to this one, This is the standard data, which comes from standard deviation divided by square root of n. And then standard data, and this whole section called margin of error. What I want to tell you here is you have a margin of error, right? So if you have plant heights 10, 10, 10, 10, you have mean 10, and do you have standard data? Your standard deviation is 0, and this entire section is 0. So there's no margin of error, so you get the exact value. But then you don't have plus or minus it. But you have values 10, 15, 12, 13, something, and then you have mean, but you have a standard deviation. So based on that, you calculate the margin of error. From that, you calculate, okay, my mean plus or minus to this section. So that's how confidence interval comes. So in reality, when you have a mean value, or if you have a value, you have to mention it with plus or minus your standard error or standard deviation, right? Some depends on the article, so depends on the test. So remember, you won't get the absolute value, the mean value, because in reality, you won't get the same numbers of your result. If you get the same numbers, maybe you run 100% theoretically correct experiment, or there's something wrong somewhere, because in reality, you won't get the same, right? That's how confidence interval comes to the picture. All right. Another simple term called central limited theorem. There's no theory behind that. It's pretty simple, although it says central limited theorem. So central limited theorem is important because if you have sample size, so basically, like I said before, your sample, if your sample size is larger, because you need the normal distributed, the Bell distributed sample results data. But if you have a larger sample size, you will get the normal distribution. But if you have a lower sample size, you may not get the normal distribution. So that's how the central limited theorem comes. It's called how you can, this is not a like a hard rule. So for quantitative variables, if your sample size is basically greater than 30, we assume that data is normally distributed, which is called central limited theorem. So central limited theorem comes to the picture based on the sample size versus whether you are skewness or not. So if you have sample size greater than 30, basically in the quantitative variable, it's normally distributed. But if you don't understand this one, don't worry about that. Right? All right. The one important thing, the normal distribution. What is that? Are there any abnormal distributions? Yeah, you already learned abnormal distribution, which are left skewed or right skewed. And this one is normal distribution. What is normal distribution? Normal distribution means the bell-shaped density curve. So it's a bell-shaped one. You will see that. And then based on that, you can, there are two main parameters in normal density curve. Your mean here, you have the mean normal distribution, and then standard deviation, which is the value deviate from your mean, explain this length. So this is your mean, and then standard deviations explain the spread. So that's how normal distribution works. And you need to convert this normal distribution, not you need to, and you convert this into a standard normal distribution. this is how they are 95%, 68 or 99% things come to the picture. So if you have your data set, you need to convert into a standard normal distribution, which is called C distribution. How do you do that? You get the mean value of your data, and then each your value minus mean and divide by standard deviation. So when you calculate this one, you get your standard normal distribution, right? That standard normal distribution is really important because that explains your the data, how you can behave. For example, here the x-axis you have standard deviations. So this is your highest value mean, and then this is one standard deviation minus plus one standard deviation, and then two standard deviations and two standard deviations. So this distribution data within these two standard deviation called your 95% of your data. So once you convert your data into a standard normal distribution, this is your 95% of your data. And then remember I told you here, this the cutoff line for rejection and non-rejection region under 95%. So this is the 5%, this plus this one called 5% rejection region. So once you calculate your value, if it falls here, it's an acceptance region, and then it comes here as a rejection region. So that's how the 95% comes to the picture. Again, if you cannot understand from this very short one slide presentation about normal distribution, don't worry. You will learn this one, or you can easily understand it. Right. So that's called Z distribution or C distribution. And then there's another one called T distribution. What is T distribution? So T distribution actually comes with the sample size. If you have a lower sample size, we usually run T distribution because T distribution addresses the lower sample size compared to C distribution. So if you increase your sample size, right, this T distribution automatically falls into C distribution theoretically, because when you increase the sample size, these two lines will fall, right? So that's the difference between C and T. But these are bit theory, but I will explain how you can use T distribution later, the T test, right? So if your sample size is smaller, you can use the T distribution, right? Now you learn a little bit of theory about hypothesis testing, and then what you need to do, and then the distribution of your data. And another important thing is, the most important thing is the test, statistical test, which you use. So one main stat test is called t-test. T-test. So t-tests are hypothesis tests that you use to analyze based on one sample, or if you have two samples. So if you have one sample, or if you want to compare two samples, so you can basically use t-tests, right? If you have one sample, we use one sample t-test. If you have two samples, we call two sample t-test, right? If you want to compare two different samples, use two sample t-tests, right? And there's another t-test. So there are three types of t-tests: one sample, two samples, and then pair. So one main thing that students always get confused is paired. cannot identify the correct paired t-test, right? So paired t-test is about how the data is the experiment in your experiment, your data is paired. I will get back to that later because I really want to explain the difference between two sample and paired t-test. So one sample t-test is a hypothesis test about population mean, and then it's a statistical test, and it's kind of a parametric test. So I put this term and put it in red because it is a bit important, parametric, the term, because there's another test called non-parametric test, right? So if your data is normally distributed, we use normally distributed mean well distributed data. You use parametric tests. So t-test is a parametric test. Or if your data is not normally distributed, you cannot use t-test. You have to use the relevant test. Relevant to the t-test, those are called non-parametric tests. I'm not going to explain this in detail because those are a bit advanced, but I just want to tell you that the difference between the parametric versus non-parametric, it entirely depends on whether your data is normally distributed or not. So that's the one sample t-test or single sample t-test, right? So one sample t-test is compared against the value, which you all know because you have one value. And then the paired compare two means that are individual or paired. So what is paired distribution? Paired is, let's say you measure effect of drug, the same person before and after. So you measure the same person, that's paired, right? So that's how it comes. So one important thing you need to understand is you have one sample, paired samples, or two samples. So then you can use t-tests, right? One sample, paired sample, or maximum two samples. What if you have more than two samples, three samples, right? More than two, what do you do? You can't use t-test, right? You cannot use t-test because t-test only supports maximum to two samples, right? If you have more than two samples, then you have to use a different test, which is called Anova, analysis of variance test, right? So analysis of variance is based on the variance analysis, but if you have more than two samples, you cannot use t-test. So that's called Anova test. So Anova compares the different groups, right? So there are various types of ANOVA: one-way ANOVA, two-way ANOVA, three-way, or repeated measure ANOVA, different ANOVA. So based on the type of the data, the type of the groups you have different types of ANOVA. All right. Right. So those are the main tests. And then another term is correlation. So what's a correlation? So correlation describes the strength of association. So correlation measures the strength of two variables. So basically, if there is an association, when you get the correlation, you get if there's a strong association or there's no association or there's a negative association. So based on that, you get the value. So the correlation value varies between -1 to +1. So -1 is the negative, strongest negative correlation. +1 is the highest positive correlation. So 0 in between, is the no association, right? So correlation comes with the correlation coefficient, which is a simple R. So that R, you will get the number, which is like 0.5 or 0.2, no, sorry, not 0.2, 0.5 or 1 or minus something, right? Or 0, 0 is there's no. So you get the larger correlation, higher the correlation is absolute value, you can say, okay, there's a strong association between two variables. But if you get the negative one, again, you can say there's a strong negative association between two variables. So these are kind of couple of correlations. So you can see this is your x-axis and this is your y-axis. So you can see dots are scattered within the x and y here. So you can see there's no association. So correlation is 0. But in this one, you can see the data goes. So this x-axis, when the x-axis goes along this way, your y goes down like this. So you can see it's very negative 0.3. So correlation 0.5. So this is a positive one. Positive one means when x increases, the y should increase. So that goes this way. So this is a negative correlation. Negative means when x increases, y goes down. So it goes like this. And then with strong correlation, you get the positive and you get the negative. That's how you get the association between two variables, and it describes with the value called correlation coefficient with the number. That's really important if you want to explain the association between two variables. Another important area is linear regression. Linear regression, it's another step of the correlation. So linear regression basically explain the distribution of your two variables, and then it fits into a mathematical model, which is called the regression model. So you have the x and y. Based on x and y data points, you plot the line of best fit, which is called this line, based on your data points, and then it makes a model, the most simple model called y equals mx plus b. You have already learned these ones. So you can see weight versus height, how it changed based on this model. So linear regression explained with the model. You can get the correlation, but correlation doesn't explain the model, but you get the regression model, and then you can explain it with the equation. So the beauty of this one is then you don't need the values in between, so you can easily predict the given x, and then you can predict the y. So if you have here, you can go back and get the y value. So that's how the model works. So there are a couple of things you need to remember in linear regression. One is slope. The slope is, this is pretty important. That's the x value. What is slope? Slope is the amount, the increase of the x if you change one single unit of x. Change of y, amount of change of y if you change the one single unit of x. So it's kind of a rate. So if you change one single, doesn't matter whether it's a kilogram, gram, but if you change one single unit of x, how much change in y? That's your x, it's slope. And then the intercept. Intercept is the value, the y value when the x is 0. If you go all the way there, when x becomes 0, and then you should have y value, that's called intercept. And then you will hear another term called residual. Residual is a value that you can calculate between a predicted value and then the actual value. So here it comes. So based on the regression model, you can get the best fit line here, this line. So this line explains all the dot. This line represents all the dots here, right, from this model. But you can see some dots are above the line, some of them are below some dots are below the line. So red dots are the below ones, blue are up. So residual means the difference. This is the actual dot and this is the model value. So difference between this and this called the residual. If you have a 0 residual, what does it mean? All the dots are on the line, right? All the dots are on the line. If you have a higher residuals are higher, that means this goes higher or this goes down. That means this gap becomes higher. That means the model, the line doesn't represent correctly. Lower the residual, higher the model value, right? So that actually, that the model whether fit or not with your data is explained with another term called coefficient of determination. Don't worry about the term. You always learn R square value, right? So R square value gives you a number that explains whether how much my line fit with the actual data. So R square value sometimes you will see with the percentage like 99%, or you will get 0.0 to 0.8, 0.9. So if higher the lower the residual, if you get the line of best fit good. So if the line explain well with the dots, you get the higher R square value, right? You always check the R square value when you do the regression that explain higher the best fit of the line. So that's a little bit of a linear regression. Sometimes you may not use linear regression. Sometimes you will use multiple regression or the different regression models, but this is the beginning of the linear regression up to even the machine learning. All right. You guys still with me, right? Right.
Yep, we're still here.
Oh yeah, I know. It's pretty boring, but I'm just talking to the monitor. Right, little bit of experimental design. We'll stop it. Just bear with me and stay with me. And Vincent is also still here. Right. So now you have I assume you have a small idea about statistics. Now let's go back to circle back to your main point called experimental designing. I'm not going to explain everything, but I'm going to tell you about main parts of your experiment and experimental design. And then we will finish the presentation. So experimental design. So you need to understand a couple of things in your experiment. One is called a factor, right? So factors are important because when someone asks or when you run a statistical analysis, most of the time, you know, what's your factor? How many factors do you have? What are the levels of the factor? So these three terms are really important. So factor is kind of a variable that you're trying to control in an experiment, right? So factors are very important. It's kind of a explanatory variable, right? So, and it should have levels. I'm pretty sure if I explain with an example, it's easy. For example, let's say effect of pesticide, particular pesticides, right? Not particular, but effect of pesticide on plant growth. So your explanatory variable is pesticide, which is a factor, right? So pesticide is a factor, right? And then you measure the plant growth, which is your dependent variable. And you have one factor, which is your, what was that? Pesticide, right? So pesticides, you have pesticides, just I don't know the chemical, right? I'm just explaining. So if you have explanatory variable, and then you should have, for example, this pesticides affect this plant growth or reduce the plant growth. So that's you have to measure. So how do you measure? You cannot use the one concentration. You use the different concentrations of the particular explanatory variable. Those are called your levels, right? So if you want to study the plant growth based on soil type and then irrigation pattern, right? Soil type and irrigation pattern effect on plant growth. So soil type is one factor and the irrigation pattern is another factor. So these are two factors. So two-factor experiment. So each factors you may have two or more levels. So different irrigation methods or different soil types. So that's how factor comes to your experiment. So each factor use different levels. So treatment, that's another term. Everyone uses treatment. So what's a treatment? So treatment is the combination, treatment is the levels of each experimental unit. So you have, for example, let's say you have two factors, so one factor, and then three levels of each. So then each level is your treatment. Or if you have two factors and each has three levels, then you should have one factor to other level, another factor, another level to another level. So likewise, you can get 6 possible treatments. So treatments are kind of the experimental, not experimental units, but kind of a combination of each factor or each level that you are using in your experiment. And then you already know the response variable, the variable that you measure. And then the important thing is the experimental unit. The experimental unit is the unit that you apply your treatment, which is this one, the combination, right? This is really important because most of the students cannot distinguish their experimental unit. This is the main part you deviate from the replication and pseudo replication. Because if you cannot understand what's my experimental unit, you will end up with pseudo replication. Right? I will get back to the pseudo replication later, but important thing is the experimental units, and remember the experimental unit is a treatment you apply. And then the replication is pretty important. Important is that each unit of the same treatment, so you call the replicate. So the replication is you supply the treatment separately, not within the treatment, because basically different experimental units. So that's called the replication. So replication is really important because higher replication reduce the amount of error in your experiment, and then that always link with your experimental unit. If you identify your correct treatments and then the experimental units, you can easily understand, okay, these are my replications. Right, let's try to understand pseudo replication. If your treatments are not replicated, or replicates are not statistically independent, you will call pseudo replication. I know it's really hard to understand, but I'll put an example here, right? So try to read and see whether you can understand the pseudo replication. So researcher studying the effect of chemical on carrot size. So you want to study the effect of chemical on carrot size. So you have one plot, carrot plot, another one is a control. And 10 carrots are sampled randomly from each plot. You have two plots, control plot and then the chemical plot. And you can measure the mean. then this becomes pseudo replication. Why is it not a pseudo replication? Because your experimental unit is a one plot. It's not, it's you collected within the same experimental unit. So that's a pseudo replication. So you should have separate plot, different plot. So that's a replication. Then you have different experimental units. Then you reduce the pseudo replication. Most of the time, we have seen students collect the pseudo replication because they cannot identify the experimental unit, and then you just collect. For example, you think you have a bigger pot on a glass house, and you have 5 plants there. You think you provide the treatment to that big pot with 5 plants, and you think when you collect the plant height, and then that's 5 different plant heights replicas. No, it's not. That's a pseudo replication. So that's why you need to understand what's the pseudo replication. There's another example. You can read it later because of the time. And then I put another one just for your information. These 3 examples, please read carefully and see whether you can understand the difference between the pseudo replication and true replication. All right. Right. Summary of today's section, not a summary, the important sections that you need to understand before you design or start the experiment. One is the randomization and the replication, and then the blocking. What is blocking? Blocking is if you have an external factor. For example, let's say there is a gradient, the water gradient on your feet. You need to block it. You need to stop it. So you block you address a blocking design. So that's another method to reduce that effect. For example, sometimes you will see in your field site, because of a big tree, you have a shade, half of the experimental site covered with a shade because you are studying the effect of something, a sunlight, but you have a shade. So then you can't cut the tree. So what you have to do is you try to block that effect. So that called blocking system. So you need to apply that one, right? So randomization, replication, blocking are important. Remember, replication, not the true replication, pseudo replication. The replication increase the precision of the estimate. So if you higher replication, you get the higher estimate, the higher value. Lower the replication, you will end up with low precision. Randomization, we already explained, and then the replication, right? Now, this slide, I'm not going to explain this link. Click this link, and then you will see different tests and what kind of analysis you want. This is exactly what you need after you collect your data. So even if you cannot understand this part, don't worry about that because me and Vincent is here to help you. So, but if you want to understand kind of a basic statistics that you want, then click this link and try to understand, okay, I have this kind of data and what kind of statistics I want. So then you have a slight idea about type of statistics you want for your study. All right. Statistical softwares, you can use different statistical software for analyze and visualize data, starting from SAS, R, Minitab, MATLAB, SAS, SPSS, I don't know, a lot of stuff there, right? But remember, if you have honor students or master student, you should know at least how to analyze your data, because most of the time students come to us with 2 weeks of the submission, say, this is my data set, what should I do? Okay, then when we say, okay, you have to do this, and then try to use one of these programs, I don't know any other program, I don't know, I haven't used it. So finally, we have to analyze your data and then you write your thesis and you don't know about your data and you don't know how to analyze your data. So it's pretty important that you should know how to analyze your data, at least if you can use at least one like R or Minitab or SPSS, that would be great. So it saves a lot of time of your study. Study means your study period, right? And then again, Microsoft Excel, I don't encourage you guys to use Microsoft Excel to analyze the data. So try to use Excel always for sorting, chart, and basic descriptive statistics, but try to avoid Excel for analyzing the data and try to use the particular proper statistical software, data analysis software to analyze your data. That makes more value because finally you need to publish this one, you need to submit this thesis. People look at this stuff very carefully. All right, the summary. Specific research question, you need to understand your research question, specify your research question that you are asking, and try to understand what's your H0, HA, null hypothesis, and alternative hypothesis, and then also type of variables and the relevant other questions, and then design your experiment with the control, randomization, everything that explained, and then collect your data, run your experiment. Once you run your experiment, you do descriptive statistics and see descriptive analysis of your data from Excel or something, and then try to understand basics or the behavior, the pattern of your data, and then run the statistical test based on your type of your data you have. And then you interpret your result and then finally communicate your results. This is the entire summary of your research journey. You have to follow these steps carefully, and then you can write your thesis. So these are pretty important. Try to follow these steps and then communicate your results effectively, right? Again, the most important thing is you need help. You definitely need help, right? Starting from your supervisor and then the senior members of your group. So these two actually, the supervisor actually help you with your experiment and all the other stuff. And then senior members gives you their experiences with the designing and everything. And based on that, you have an idea about your design and everything. But if you want to understand more, or if you want more elaboration about your experiment design, or if you want to know about your design and how to analyze them, and if you want data, any help, statistical analysis, anything, you contact me or Vincent. This is our email address, biometrics1@uq.edu.au. Contact us. I'm in St. Lucia, Vincent is in Gatton. We are here to help you. So if you have any question, don't hesitate to contact us and make an appointment, and we are here to help you, right? Not only about that, even for software help, we are here to help you. And then one small piece of information, if you go to UQ Library, there's another link called lynda.com. If you go to lynda.com, you can get some idea about some special training about the Excel or R or any statistical program. If you are really interested in mastering one program, that's a pretty good free platform. But anyway, we are here to help you. All right, thank you so much. That's the end of our, how long did it take? Nearly 2.5 hours of everything, like 10 weeks of stat. I tried to explain in 2.5 hours. Thank you very much for listening. You're still here with me.
Thank you, Buddy and Vincent. That was really good.
Thanks, guys. Yeah, appreciate it.
All right, so I will upload this presentation on Blackboard. If you guys want, you can follow. Yeah, any other questions, Vincent, in the chat?
Nope. No, we're
All good?
good. Yep.
All right, so this is our email address again. If you want, you can contact us even before you start experiment, because once you, if you come with the data set 2 weeks before your thesis submission, I'm talking with our experience, we just can give you the postmortem of your data. So don't do that. Come and meet us. All right. So good luck with your research. See you guys.
Thanks.
buddy.
Bye.
Love your work.
See you guys. Thank you.
Yeah.